You begin by letting your breath settle into a slow, steady rhythm, a quiet tide that rises and falls without hurry. Each inhale draws you a little deeper into yourself, and each exhale softens the edges of your awareness. As your body loosens, you sense a gentle doorway opening inside you, an invitation to explore the delicate architecture that has supported your life since your first moment. You drift inward, not with urgency but with a calm curiosity, allowing the landscape of your inner world to come into focus. In this softened space, you imagine your body as a vast, peaceful city made of countless small rooms. These rooms are your cells, tiny sanctuaries humming with structure and purpose. You picture them glowing softly in the dimness, arranged with astonishing care, each one shaped not by chance but by a quiet, ancient logic. Your attention lingers on the graceful variety of these rooms. Some designed to carry oxygen like gentle couriers, some built to sense the world, some constantly repairing, some quietly remembering. And in every single one, the same essential blueprint lies waiting. As you float deeper, you approach one room and slip inside. You find yourself in a space so small you couldn't see it with your eyes alone. Yet here it feels expansive, almost like a cathedral made of living matter. In the center is a coiled structure, delicate and intricate, resting like a scroll written in languages older than memory. This is DNA, the set of instructions your body reads every moment, the blueprint that guides every gesture of life within you. You approach it with reverence, sensing the quiet power held in its loops and turns. The strands of DNA feel almost alive in your imagination, shimmering like threads woven from moonlight and intention. Their shape, the familiar double helix, rises like a staircase turning endlessly upward. Each step holds information, and your body, without conscious thought, walks the staircase constantly. Proteins move along the strands like librarians, opening sections, copying messages, passing them along to where they're needed. This is not a frantic process. It is measured, steady, and deeply precise, an act of constant caretaking that unfolds within you even now. You notice how these messages, written in sequences of chemical letters, tell each cell what to do and when to do it. They guide growth, healing, renewal. They tell immune cells how to recognize what belongs to you and what does not. They help your skin mend, your muscles strengthen, your memories form. It is astonishing to realize that while you rest, while you breathe, while you lie here listening, this silent reading continues without pause. You sense the intimacy of it. Your body is not a machine acting at random. It is an orchestra tuned by the instructions in these spirals. Each note, each movement, each response to the world is shaped by the language written inside you. And yet, despite the elegance of the design, you feel how fragile and flexible it all is. A small change in the sequence can shift the meaning of a message, just as a misplaced word can alter the tone of a poem. You drift from cell to cell, sensing how this architecture repeats with variation, forming tissues, organs, systems. The heart beats because certain cells follow certain instructions. The lungs expand because others do. The immune system stands ready because entire networks know how to read the messages assigned to them. Everything depends on this continuous conversation between the written code and the living body. You pause to appreciate how your body protects this information. DNA is kept safe inside the nucleus, wrapped around proteins like a treasured manuscript bound in careful covers. When damage happens, as it inevitably does, repair systems move in, checking for mismatched letters, restoring the spiral to its proper shape. Even at rest, you are never truly still. Your cells are always tending, always maintaining, always preserving the integrity of the script. This awareness brings a gentle warmth, a sense of gratitude for the quiet labor happening within you. You begin to understand the elegance of immunity not as a shield held against the world, but as a series of responses guided by the architecture you are now drifting through. The ability to recognize a virus, to calm inflammation, to heal after illness. All of it begins here in these spirals of instruction. As you explore, a soft wonder forms. If these messages guide immunity so deeply, then what would it mean to adjust them? To revise a single line? To strengthen a phrase that helps the body recover or quiet one that leads to overreaction? You don't rush toward an answer. You simply let the question float beside you, like a lantern suspended in still water. You sense that before such possibilities can be understood, you must first feel the foundation beneath them. The blueprint, the alphabet, the quiet architecture of your cells. This is where the story of immunity begins. And here, in this inner landscape, you feel the first hints of how the narrative might someday be gently shaped. You rest in the glow of these thoughts, sensing the spirals around you, sensing your breath anchoring you to the present moment. The city of your body feels vast again, stretching outward from this tiny room, filled with countless other sanctuaries carrying the same quiet script. And as you drift toward the threshold of this chapter's end, you begin to feel another question rising softer still. If the instructions inside your cells shape your immunity, then where did the first immune instructions come from, and how did life learn to write them at all? You allow yourself to linger in this question, letting it unfold slowly in your mind. The air within the imagined cell feels gentle and warm, as though it, too, is waiting for you to consider the depth of what you have seen. You picture the earliest cells on Earth, floating in ancient oceans, carrying simpler instructions, simpler protections. You imagine how, over unfathomable time, these instructions grew more detailed, more refined, more attuned to the challenges of life. It feels almost like watching a long-forgotten story rediscover its voice. As you continue drifting, you sense how the architecture within you is both ancient and new. Every moment your cells live, they renew parts of themselves, rewriting tiny sections, repairing what is frayed, adjusting to new needs. Yet the core of their blueprint remains tied to a lineage stretching back billions of years. You feel yourself connected to that lineage, part of an unbroken thread of life learning how to endure. This thought settles over you like a soft blanket. Your body is not only a structure but a history, a collection of remembered solutions to the challenges faced by all the organisms before you. And immunity, you realize, is one of those deep solutions, a system built layer by layer, refined through time, encoded in the spirals you have just explored. You let this realization breathe within you, slow and calm, knowing that understanding these foundations will help you follow the story as it continues to unfold. You pause on this threshold, knowing the next step will lead you further back in time, toward the ancient origins of immunity itself. You begin by drifting back through time, carried on a slow current of curiosity that feels as gentle as the breath moving through your chest. You let your awareness loosen, softening around the idea that long before you were here, long before complex bodies or tissues or organs existed, life was already learning how to protect itself. You imagine yourself gliding downward through the layers of history, slipping past eras and extinctions, until the world becomes very simple, very small, and very still. In this quiet scene, you find yourself beside the earliest bacteria, tiny beings floating in primordial waters. They are humble, unassuming, almost invisible in their simplicity, yet they hold the beginnings of something profound. You sense their vulnerability, their openness to the unpredictable touch of the environment. You notice how, even in this simplicity, survival requires awareness, memory, and response. These tiny organisms have no nervous systems, no organs, no conscious decisions, and yet they behave with an elegant kind of intelligence shaped entirely by nature. You watch them drift, each one like a small bubble of life containing the essential machinery to grow and divide. Their world is not peaceful. It is filled with invisible threats. Among the most dangerous are viruses, bacteriophages, that land on their surfaces like small, sharp seeds and inject their own genetic material within. For many bacteria, this intrusion is devastating. A virus can take over the cell's internal processes, forcing it to produce copies of the invader until the cell bursts, releasing more viral particles into the water. You sense the fragility of this early life, but you also sense its determination. Evolution is patient. Over countless generations, bacteria begin to develop a remarkable strategy, a way to remember the viruses that threaten them. It is here, in this ancient ocean, that CRISPR quietly begins. You imagine floating even closer, watching the molecular world unfold, as if illuminated by a soft inner glow. You see the CRISPR sequences take shape inside the bacterial DNA, short repeats of genetic letters, arranged like rhythmic patterns in a whispered chant. Between these repeating sections, you notice unique fragments, each one captured from a virus that once attacked the cell. These fragments are memories, written in the language of life. CRISPR, you realize, is not a tool humans invented. It is a natural archive that life created for itself. You feel the poetry of it. An organism so small it cannot be seen with the naked eye carries a library of its past threats, stored not in stories or drawings, but in sequences of nucleotides. When a virus invades again, the bacteria draw upon these memories, using them as guides to recognize and cut the matching genetic sequence. It is a defense that feels almost aware, almost intentional, though it arises simply from the elegant logic of molecular interactions. You drift along this ancient timeline, imagining how this system evolves. Proteins develop to work alongside the CRISPR sequences, Cas proteins, which act like careful guardians. They search quietly for anything that resembles the viral fragments stored in the bacterial memory. When they find a match, they cut it swiftly, disarming the threat. It is a choreography as old as life itself, a dance of recognition and response. You feel a sense of awe at how such a tiny organism can carry such profound capability. This is immunity in its earliest form, a memory system without consciousness, a defense system without anger or intention, only clarity and precision. It teaches you that protection is not always about strength. Sometimes it is about remembering, adapting, and responding with quiet accuracy. As you continue to drift through this molecular world, you picture the passing of eons. Bacteria replicate, evolve, diversify. Their CRISPR systems grow more refined, more efficient. They hand these tools down through generations, shaping lineages that spread across the planet. Long before humans appear, long before animals or plants or even complex cells take form, this ancient technology is already in motion, quietly solving the problem of survival through memory. You pause to let this understanding settle. You realize that the concept of editing genetic material, something humans only recently learned to imagine, has existed in nature for billions of years. The bacteria are not engineers, and yet their genomes hold an editing toolkit. They do not plan, and yet they adapt. You feel a deep, slow gratitude for the long journey life has taken to create the tools we now study with intention. You float gently above this whole history, watching the unfolding transformations with a sense of calm admiration. You understand now that CRISPR is not a modern miracle, but an ancient wisdom, one that nature refined long before human science gave it a name. And you feel your thoughts shifting softly toward a new question, one that rises like a quiet ripple. How do these ancient tools become the gentle molecular hands we now call Cas proteins, and how do they guide the genome with such steady, deliberate care? You let your awareness drift, resting in the ancient rhythm of these early defenses as the story continues to unfold around you. You begin this chapter by letting your breath slow, allowing it to steady the gentle rhythm that carries you inward. You imagine yourself drifting once more through the microscopic world you have already touched, returning to the hidden place where ancient memories float inside living cells. You feel as though you are walking into a quiet workshop, a space shaped by patience rather than urgency. And in that space you sense that calm molecular hands are waiting for you. You move deeper, weightless, as strands of DNA shimmer like soft threads suspended in the air. Their repeating patterns shine with a slow, comforting pulse, as if they are aware of your presence. Then you sense movement, subtle at first, like the faint lift of a curtain in a still room. A Cas protein approaches, not rushing or pushing, but drifting with a quiet confidence that feels almost reassuring. It has no eyes, no face, yet you can feel its attention, its readiness to respond. The Cas protein carries a ribbon of RNA, a memory folded into form. This ribbon guides it the way a friend might gently lead you through a dim hallway, offering direction without force. You picture this RNA whispering coordinates, its message soft but certain. The Cas protein listens in the only way molecules can, by shape, by touch, by resonance. It turns as though following a familiar melody, its motions smooth and practiced. As you float beside it, you watch the protein search the DNA for a matching pattern. You imagine it scanning like fingertips brushing over braille, feeling for a sequence that calls to it. The moment it finds the match, everything stills. The protein pauses, suspended in a hush that feels almost sacred. Then it reaches out, touching the DNA with careful precision. There is no tension in the motion, no sudden snap. Instead, the Cas protein moves like an artisan making a clean, thoughtful cut in a delicate piece of paper. You see the strands separate gently, the harmful viral sequence released like a loose thread pulled from a fabric. The protein's task is simple but essential. Protect, preserve, maintain balance. You sense its purpose the way you sense the weight of a stone in your hand. Quiet, steady, fundamental. And even though you know these actions arise from chemistry and evolution, the moment feels intentional, almost tender. You drift alongside the Cas protein as it moves away, carried once again by the soft guidance of its RNA companion. You begin to notice other Cas proteins in the distance. Some glide slowly, as if mapping the space around them. Others rest, folded neatly, waiting for a cue only they can detect. They feel like gentle workers in a vast, molecular garden, tending to each plant with subtle, practiced gestures. As you watch them, your understanding grows more textured. You realize that these proteins are not all the same. Some specialize in cutting, others focus on guiding or binding. Some lift away harmful fragments, while others act like careful scribes, helping the cell remember what it has endured. Their roles form a quiet orchestra of responses, each voice distinct, each movement contributing to the larger harmony of immunity. You imagine tracing one protein's shape with your mind's fingertips, feeling the grooves and curves carved by evolution. Its structure is complex, yet somehow peaceful, like the surface of a stone shaped smooth by centuries of passing water. You sense that its beauty lies not just in its appearance but in its purpose, in the way function and form fuse naturally together. You stay with this image as the proteins continue their work around you. Their motions feel almost meditative, as though each small action echoes a larger truth about resilience. You begin to see how scientists, observing this natural process, learn to use these tools to edit genes in plants, animals, and people. What began as a quiet bacterial defense became a doorway into new possibilities. And even though those possibilities carry responsibility and caution, there is a feeling of hope woven into the edges of the story, a softness that suggests potential without pressure. You breathe slowly, letting that hope settle within you. You consider how something so tiny can hold such potential, how these molecular hands might someday help guide healing in deeper, more intentional ways. Their calm movements seem to whisper that change does not always have to be abrupt. Sometimes it can be slow, thoughtful, and guided by memory. As you drift, you feel the story widening, pulling you toward a larger landscape. If Cas proteins are the gentle hands, you wonder what the whole organism feels, the broader system these hands support. You sense the question forming like a soft glow on the horizon. What happens when these small actions scale into the sweeping patterns of immunity itself? You let the quiet images settle, feeling the soft continuity between what you have learned and what still waits to be discovered. You begin by letting your breath settle into a slow, even rhythm, allowing it to guide you once more into the inner landscape where stories unfold without sound. As you drift inward, you feel yourself entering a place where your body is both narrator and listener, shaping a tale written in countless small gestures. This is the place where immunity becomes a living story, one that moves through you with quiet purpose, always watching, always remembering, always adapting. You picture the vast interior of your body not as machinery but as a soft, shifting terrain of motion and conversation. Cells move like characters wandering through a shared world, sometimes alone, sometimes gathering in small constellations of purpose. You sense the gentle pulse of chemical messages flowing between them, each one carrying meaning the way a whispered phrase might carry comfort. These signals are not loud. They do not shout. They drift like glowing threads, weaving responses that feel almost musical in their timing. You follow one of these signals as it glides through your bloodstream, drifting toward the cells that wait like attentive listeners. When the message arrives, the cells respond not out of fear, but out of readiness. You notice how their actions feel like the turning of pages in a quiet book. Deliberate, calm, unfolding with a rhythm that makes sense only when you slow down enough to witness it. You begin to see that immunity is less about battle and more about balance, more about conversation than conflict. As you move deeper, you encounter macrophages, the large, steady guardians that roam your tissues. They feel like storytellers, gathering fragments of what they encounter and offering them back to the system as lessons. When they find something unfamiliar, they do not panic. Instead, they lift it gently, as though examining a curious object washed ashore. Then they bring it to other cells who gather around like attentive students, learning from what the macrophage has found. The story grows richer with every shared detail, every exchanged signal. You wander farther and watch T cells drifting like thoughtful scouts. Each one carries a memory shaped by past encounters. They move with purpose, guided by patterns they have learned. You imagine them as readers who know how to recognize a familiar chapter even when the details change. Their role feels like interpretation, seeing meaning where others see only shapes. When they sense something troubling, they do not erupt. They respond with measured clarity, contributing their insights to the ongoing narrative of defense. Nearby, B cells rest like quiet archivists. They hold the potential to craft antibodies, the small messengers that move through your body with the devotion of emissaries delivering protective notes. When the moment calls for it, B cells begin writing, shaping molecules that fit threats as precisely as keys fit locks. Their work feels like transcription, copying the lessons the body has learned into forms that can move freely and help maintain harmony. You sense their patience, their restraint, their willingness to wait until the story requires their lines. As you watch these cells, you realize that immunity is not a single story but 1,000 overlapping tales. Some are short, ending within hours. Others stretch across years, forming a deep memory that shapes how your body responds long into the future. You drift through these stories and feel their presence like a gentle echo. Every encounter becomes part of you, written quietly into the chapters of your body's history. You take a slow breath, absorbing the idea that your immune system is not merely reactive but deeply perceptive. It learns, it remembers, prepares, and through all of this, it maintains a delicate equilibrium, adjusting itself with every new experience. You imagine the system as a river, always moving, always reshaping its shoreline as fresh currents flow in. Nothing is static. Everything is becoming. You feel a soft curiosity rise within you as you drift along this river of responses. What would it mean to guide this story with precision? What would it mean to help the immune system learn more clearly, more gently, more efficiently? You picture the possibility like a lantern glowing faintly ahead, illuminating the next bend in the river without revealing everything at once. You return your attention to the present moment, noticing how calm permeates every part of the system when balance is maintained. You sense how gratitude might feel if cells could experience it, the gratitude of being part of a living, interconnected whole. Each cell plays a role, but none acts alone. Signals ripple outward, and the entire system listens, responds, adapts. As you continue drifting, you reflect on how scientists have begun to understand this living story with greater clarity. They observe how cells share information, how they remember past intruders, how they adjust their strategies with each new encounter. And through these observations, new possibilities emerge, possibilities that hint at the potential to gently rewrite the narrative when needed, to help the immune system learn new lessons with care. You remain suspended in this quiet understanding, feeling the story of immunity unfolding around you. It is not a tale of constant struggle but one of subtle negotiation, of harmony regained again and again. You sense that this story is still being written, shaped by every breath you take and every moment you live. You begin this chapter as though stepping into a quiet rehearsal hall within yourself, a place where silence feels expectant rather than empty. Here, your immune system is not a battlefield but a gathering of instruments, each tuned by the subtle pressures of time and experience. You imagine these cells not as warriors, but as musicians carrying memories in their structure, adjusting their melodies to your shifting inner world. And tonight, you explore how gentle scientific guidance can help those melodies find new clarity, new ease, new harmony. CRISPR enters this space as a calm presence, not a tool of force but a guiding hand. In this chapter, you walk slowly beside the idea that editing immunity is less about control and more about resonance, about listening for the notes that want to be shaped. The scientists who study these processes often describe them in technical language, but you sense a deeper story beneath the terms, a story of collaboration between human curiosity and nature's ancient creativity. As you settle deeper into the imagined hall, you picture immune cells drifting like lanterns in a dim, warm air. They glow with quiet purpose, always listening for signals that travel through your tissues. Some respond to injuries with urgent brightness. Others remain soft, waiting to be called upon. Together, they form layers of protection that shift depending on your environment, your memories, and even your sleep. You feel the fluidity of this system, its responsiveness, its constant improvisation. Now, CRISPR moves into the scene like a conductor stepping onto a low-lit stage. But this conductor does not impose rhythm. Instead, it offers suggestions, tiny adjustments that help each instrument find its clearer voice. You imagine the guide RNA, a thread-like whisper, floating gently toward a cell, carrying a memory of a particular sequence. It is not a map for destruction. It is a note of recognition, a reminder of where attention might be directed. You watch as this whisper settles near strands of DNA, though you keep the details soft, staying within the poetic landscape. You do not follow the mechanism with clinical precision. Instead, you follow the feeling of alignment, the quiet miracle of recognizing something familiar in a vast sea of information. The Cas enzyme you met earlier moves like a steady, careful hand, pausing before making its subtle mark. It feels less like editing and more like tuning, tightening a string, loosening another, helping the music settle into smoother chords. Scientists imagine many possibilities when they look at your immune system through this gentle lens. They wonder whether certain immune cells could be guided to respond less intensely, creating more calm where inflammation once roared. They envision helping other cells recognize patterns they previously overlooked, adding new notes to their repertoire. And although these ideas are still emerging, you sense the beauty in their intention. The desire to shift from blunt interventions to something far more precise, far more compassionate. You imagine one immune cell floating close. It has traveled through countless reactions, carrying traces of past fevers and fleeting illnesses. Tonight, though, you let it drift without urgency. CRISPR's presence approaches not with command but with a soft invitation to adjust, to consider a gentler path. The metaphoric tuning fork hums, and the cell listens. Its internal symphony shifts almost imperceptibly, like the moment an orchestra warms into harmony. As you observe this, you realize that your immune system is not fixed. It is a living story, changing with each breath, each exposure, each night of rest. CRISPR simply offers a new way to participate in that story. The edits are not brute changes. They are explorations of possibility, attempts to understand how each note influences the whole piece. You imagine scientists sitting in quiet labs late at night, the dim hum of equipment surrounding them. They are not so different from you, listening to the rhythms of your body as though it were a distant symphony. Their curiosity is tender, grounded in a desire to understand rather than to dominate. You sense their awe when they watch immune cells shift behaviors, learning from tiny sparks of information introduced with care. It feels like watching seeds sprout under gentle hands. Your attention returns to the rehearsal hall. More cells gather, glowing softly. Some carry instructions that allow them to calm inflammation. Others help recognize lingering threats. You imagine CRISPR helping them communicate more clearly, smoothing misinterpretations that sometimes cause your body to overreact. And though these images remain metaphorical, they reveal the essence of the science. The hope that one day immune responses might become more harmonious, more attuned to what your body truly needs. You begin to feel the weight of this harmony. It spreads through the imagined space, settling across your inner landscape like a warm, balanced breeze. Each cell seems to breathe in unison, listening for the subtle cues that guide its actions. You sense how intricate this world is, how many layers of coordination unfold without your awareness. And here, CRISPR becomes a quiet collaborator rather than a force of change. A gentle suggestion in the larger conversation that keeps you alive. You walk deeper into this world, letting the metaphors unfold without rushing. A cluster of immune cells gathers like a small ensemble. They have been responding to inflammation for longer than they remember, flaring brightly at the slightest signal. Tonight, though, a softer possibility enters their space. CRISPR murmurs through the guide RNA, offering a way to lessen the urgency, to create space between stimulus and response. The cells begin to practice new patterns, the way an ensemble considers a different rhythm, discovering a more measured pace. Further ahead, another group of cells glows with a different purpose. They are the recognizers, the ones who listen for molecular signatures of things that do not belong. You imagine CRISPR gently adjusting their sensitivity, not to make them aggressive, but to allow them to read the world more clearly. The metaphorical tuning fork hums again, helping these cells refine their ability to interpret signals without confusion. As you follow these scenes, you understand that this chapter is not about modification for its own sake. It is about possibility, about the idea that even your defenses can learn to be more compassionate, more precise, more in tune with the rhythms of your life. CRISPR becomes a reminder that change does not always need to be dramatic. Sometimes the smallest adjustments create the most profound shifts. You take a breath, imagining your entire immune system inhaling with you. The hall brightens slightly, illuminated by the glow of countless cells finding new balance. The melody they create is subtle, almost imperceptible, but you feel it nevertheless. A hum of potential that moves through your chest with a comforting warmth. Before you step fully away, you pause in the quiet doorway of the hall, sensing that something still wants to be acknowledged. There is a recognition that harmony is not a single moment but a practice, a continuous attunement that unfolds with each new experience. In this stillness, you sense your immune symphony learning how to breathe in new patterns of balance and patient transformation with gentle intention. As you prepare to leave this chapter's hall, you listen to the lingering resonance. It stays with you, vibrating softly in the background of your awareness. CRISPR has shown you how gentle guidance might shape the stories within your body, how new harmonies might emerge from careful attention. You drift into a quiet realm where memory is not a single place but a series of soft chambers, each one shaped by encounters your body has weathered. Here, immune memory is not a rigid archive but a breathing presence, warm and shifting, like light moving across the floor of a dim room. You feel yourself wander through these inner corridors, noticing how the stories of past invaders glow faintly, held not in words but in molecular gestures. And as you step softly, you sense that these memories are always learning, always being rewritten by subtle forces you barely notice in waking life. CRISPR enters this imagined space like a calm guide, not here to erase or override, but to suggest new forms of remembering. You picture it as a lantern carried gently through the corridors, illuminating patterns that once felt fixed. The glow shifts the way your immune cells interpret old lessons, offering them a chance to loosen or strengthen depending on what will serve you best. You begin to see how memory is more fluid than you once believed. Not a locked story but a living process shaped by every breath, every season, every quiet healing. As you walk deeper, you notice a cluster of memory cells resting like small, attentive birds. They hold fragments of past challenges, poised to respond if those challenges ever return. Yet tonight, they seem peaceful, listening for new guidance. CRISPR stands beside them metaphorically, offering a gentle adjustment to the way they store their insights. You picture the shift not as a sharp change but as a soft recalibration, like retuning an instrument so its notes carry more clarity, more ease. You feel the weight of this metaphor settling inside you, that your immune memory is not a harsh sentinel but a thoughtful observer. It learns, it adapts, it remembers not to cling, but to protect with understanding. CRISPR's presence highlights this natural wisdom, showing how small adjustments can refine the balance between vigilance and calm. Some memories may deepen, becoming clearer guides. Others may soften, releasing old tensions your body no longer needs to hold. In another chamber, you encounter memories formed from moments of inflammation, times when your body reacted with more urgency than was necessary. These memories hum with a restless energy, still echoing with the intensity of their origins. Yet CRISPR drifts close like a quiet breeze, offering new possibilities. It suggests a way for these memories to loosen, to unlearn the reflex of overreaction. You sense a gentleness in this process, a compassion woven into the very idea of editing. It feels like smoothing the rough edges of a story that has weighed on you for too long. You move on, finding cells that have learned from viral encounters long past. Their memories glow with a steady confidence, ready to respond if needed. CRISPR's presence here is reverent, acknowledging the strength these cells carry. Instead of reshaping, it simply clarifies, helping the cells distinguish old threats from those that resemble them only faintly. In this way, memory deepens without becoming rigid, remaining flexible enough to understand what is truly present and what is merely similar. As you wander, you notice how these adjustments, these metaphoric touches, change the atmosphere of the chambers. The memories feel lighter, more attuned, less burdened by old intensity. You sense how your future immune responses may shift because of these softer recalibrations. The future is not yet here, and yet its shape changes subtly through the ways your cells prepare. This awareness settles warmly in your chest. The understanding that immunity is not only about reacting, but about learning how to respond with wisdom. You pause in a chamber filled with a golden, drifting haze. This is where naive cells wait, cells that have not yet learned their roles, untouched by past encounters. CRISPR stands near them like a teacher offering quiet introductions to the world they will someday face. Not instructions, but possibilities. You sense the beauty in this, that learning can begin before experience, that preparation can be shaped with care so future memories form with balance rather than fear. The haze swirls gently around you, and you realize how vast this inner terrain truly is. Immune memory spreads across countless cells, each holding a small piece of the story. Some memories protect you fiercely, others guide more subtly, and all of them contribute to the way your body meets the world. Tonight, you let yourself feel the vastness of this coordination, the countless interactions that unfold without your awareness, keeping you alive in ways both quiet and profound. CRISPR remains a soft presence as you walk. It does not dominate these chambers. Instead, it harmonizes with them, offering a way to refine the narrative held in your cells. You sense that its true power lies not in what it can change, but in the gentleness with which it can help your body learn differently. It becomes clear that editing immune memory is not about creating perfection. It is about guiding patterns toward greater balance, helping future responses unfold with more clarity and less strain. You step into a chamber where memories of childhood illnesses linger. They drift like faded tapestries, their colors muted but still present. CRISPR walks beside you, acknowledging these old stories with a soft glow. Some memories remain important, shaping your resilience. Others may be outdated, holding on to echoes that no longer serve. You watch as the light subtly shifts these memories, refining the lessons they carry. Not erasing experience, but transforming it into something more aligned with who you are now. Farther on, the chambers grow quieter. These are the memories still forming, shaped by recent encounters or subtle shifts in your environment. You feel yourself walking through a landscape of near-present moments, sensing how your body learns continuously, even when you are unaware. CRISPR moves gently here, listening, observing, offering only the lightest of suggestions. It feels like watching a conversation unfold between different layers of yourself, a dialogue rooted in patience, curiosity, and care. All around you, immune memory becomes less like a static archive and more like a meadow in constant motion. Winds shift, seeds disperse, new growth emerges, and old growth fades into nourishing soil. CRISPR's presence is like a gardener's touch, refining the landscape without dominating it. You sense how each small adjustment influences the seasons to come, altering the future with the softest hand. As you begin to leave the chambers, you pause once more. The memories hum around you, vibrant and balanced, shaped by both experience and gentle guidance. You sense the story of your immunity unfolding ahead, not as a rigid script but as a living narrative, learning and adapting with every encounter. You move through quiet laboratories where edited immune cells are grown with care. Each step feels deliberate, slow, and hopeful. You imagine cells returning to the body like familiar companions, better prepared to protect you. A soft horizon opens, a future shaped by careful, compassionate science. You breathe in the hush of the laboratory, feeling the quiet weight of possibility. Cells rest in warm light, drifting like tiny moons in gentle orbit. Scientists move with soft intention, their motions shaped by patience. Each edited cell carries a subtle shift, a quiet turning toward healing. You imagine these cells returning to the body, moving through you like familiar whispers. CRISPR becomes a steady hand, offering guidance inside the living story of immunity. You sense how the future unfolds through careful steps, never rushed. The promise of therapy hums like a distant melody, calm and low. The immune system listens, learning new patterns with each gentle change. You feel held within this unfolding, as if the science itself breathes with you. This work is slow, deliberate, shaped by respect for what the body already knows. Edited cells become companions, carrying new clarity into old pathways. Hope forms in quiet layers, never sudden, always growing. You watch as understanding deepens, forming bridges between intention and biology. Light settles on glass vessels, and the room feels like a sanctuary. Every adjustment is a soft tuning, preparing harmony within the body. Possibility moves like water, steady and continuous. Here, healing feels like a conversation spoken in molecular tones. CRISPR's precision becomes a promise of gentler futures. You drift with these thoughts, letting them settle into stillness. Hands in the lab handle each sample with a reverence that resembles prayer. Technicians whisper to instruments as if to coax good outcomes into being. Under magnification, cells appear as communities practicing a new language. You sense the tenderness in procedures that honor life at its smallest scale. Edited immune cells learn to listen rather than to overreact. Each small correction can ripple outward, changing patterns of response. The idea of therapy becomes less about replacement and more about restoration. These therapies help the body remember how to be at ease with itself. You imagine cells that once misread signals now recognizing nuance. The lab becomes a place of patient craftsmanship, not haste. Researchers chart each step carefully, listening for unintended echoes. They tweak, observe, and wait, favoring slow confidence over quick fixes. Protocols read like gentle instructions for a delicate revival. In culture dishes, cells practice new roles until the behaviors feel natural. You feel the slow accumulation of care across these attentive hours. When the time is right, edited cells are prepared to return home. The process of rain fusion is like a quiet ceremony of reunion. Cells re-enter the bloodstream and travel toward tissues they know by memory. They carry their edits like new stories learned on a careful journey. As they settle, they offer their revised skills to the community of the body. You imagine inflammation softening like a storm slowly losing intensity. Conversations that once erupted now take on a measured tone. Where confusion once sparked flare-ups, recognition grows steadier. You sense the relief that comes from a system learning subtlety. Therapies like these are not magic. They are patient acts of refinement. They honor complexity rather than trying to flatten it into simplicity. You feel gratitude for the humility of scientists who respect living systems. They seek not to dominate but to collaborate with the body's intelligence. The laboratory's calm reflects a commitment to safety and compassion. Data are gathered meticulously, each result a soft footprint toward truth. Clinical teams frame each outcome as part of an ongoing conversation. Patients and cells alike become partners in an unfolding experiment of care. You imagine family members watching with cautious hope as therapies progress. Stories of small improvements spread gently, like lanterns in the dark. Medicine shifts from abrupt intervention toward thoughtful accompaniment. Edited immune cells can be tailored to fight cancer with renewed focus. You picture cells trained to recognize malignant patterns with clarity. They patrol tissues with purpose, distinguishing damaged from healthy. These therapies carry a tenderness, to correct, not to destroy indiscriminately. In autoimmune conditions, edits can help cells quiet their mistaking of self for foe. You sense how relief might wash over those long burdened by chronic inflammation. The possibility of easing suffering sits quietly at the heart of the work. Still, the path is measured by caution and ethical reflection. Scientists discuss risks and benefits like elders weighing important choices. Each trial is built with oversight and a slow attentiveness to consequence. Regulation and care intertwine, ensuring that curiosity is balanced by safety. You sense the moral landscape as broad and luminous, requiring steady hands. Communication in the lab is soft but clear, filled with questions rather than certainty. Researchers document not only outcomes but the questions that remain unanswered. This humility becomes part of the culture, a protective habit of mind. You imagine communities participating in dialogues about how to use these tools. Public trust grows when conversations are open and intentions transparent. CRISPR therapies are shaped by science and by the values that guide their use. You feel a larger social fabric woven into each clinical protocol. The work in the lab ripples outward to policy meetings, patient councils, and classrooms. Education becomes a gentle bridge between possibility and understanding. Through teaching, communities learn how to weigh hope with prudence. You imagine future clinicians trained to listen with equal measures of skill and empathy. In hospitals, edited cells are delivered with rituals of care and precise timing. Nurses and doctors watch for subtle signs that the therapy integrates smoothly. The return of edited cells is celebrated quietly as patients rest and recover. Families notice small changes. Less pain, more ease, a deeper sleep. You sense how these small shifts build a mosaic of lived improvement. Over time, the cumulative effect becomes a new pattern of health. Research teams continue to follow outcomes with long, attentive studies. They trace how edits alter life trajectories across months and years. Data becomes a story told in living detail rather than a single snapshot. You feel the unfolding of hope, steady and cumulative, rather than sudden. The promise of CRISPR therapies rests on patience, care, and continual learning. It asks us to reimagine medicine as collaborative rather than conquering. You imagine a future where treatment and tenderness coexist naturally. This image settles into you like a warm light, steady and generous. You begin this chapter drifting into a quiet inner landscape, one that feels both familiar and surprising. It is the terrain of your own immune system, a world that lives within you, working every moment without asking for recognition. In this stillness, you sense the way its movements resemble weather, sometimes calm, sometimes stormy, always shifting. You have not thought of it this way before, but now the idea opens slowly. Your immunity is not war, it is balance. A constant, gentle weighing of signals, a negotiation between activity and rest. You breathe into that discovery, letting it settle like fine dust across your mind. As you continue to float through this inner world, you notice how delicate the equilibrium truly is. Your immune cells must decide again and again what is harmful and what is harmless. It is not easy. They listen for clues, whispering chemical messages, passing information like small lanterns from hand to hand. When everything works smoothly, your body thrives quietly. You hardly notice the careful choreography unfolding inside you. But sometimes, a misstep occurs and the dance falters. The very cells meant to protect you become unsettled, uncertain, almost distrustful. They begin to respond to things that are part of you, or to signals that were meant to be soft but are interpreted as sharp. This is where autoimmunity begins, an orchestra that plays too loudly, too long, or in the wrong direction. In this meditative moment, you imagine yourself observing the cells from a distance, watching them with compassion rather than fear. They are not trying to harm you. They are confused, as if seeing through fog. You sense how comforting it is to view them this way, not as enemies, but as overworked guardians who need guidance, rest, or clarity. The body is always trying to heal, even when its efforts go astray. That thought alone brings a quiet warmth into the scene. You drift deeper, noticing how scientists have come to understand autoimmunity as a kind of imbalance. Not failure, imbalance. Signals that should fade linger instead. Messages meant to soothe become amplified. Cells trained to recognize self, forget their lessons, and turn back to questions they once answered confidently. It is like a memory slipping, or a note in a familiar song suddenly sounding out of tune. You can almost hear it, a slight discord that expands if left unattended. But then, with a gentle shift, CRISPR enters the picture. Not as intervention, but as careful tuning. You picture it as a musician stepping into a quiet hall, holding not a loud instrument but a small, precise tool. CRISPR does not shout. It listens. It studies the genetic instructions inside immune cells, like reading sheet music that has been copied and recopied through generations. And when it finds a note that has been written incorrectly or has begun to create confusion, it can make a change so small you would hardly believe it could matter. Yet it does matter. A single corrected note can restore harmony to an entire symphony. You let yourself imagine these adjustments as strokes of calm light across the genome. Perhaps a receptor on a T cell is tuned to be less reactive. Perhaps an inflammatory signal is softened, like lowering the flame beneath a pot that has begun to boil too vigorously. Perhaps a regulatory cell is strengthened, given the ability to speak up and gently hush its overactive neighbors. These images float like watercolor, delicate and slow, each one revealing how balance can be shaped rather than forced. As you drift further, you realize that CRISPR's promise is not to erase the immune system's power, but to restore its wisdom. Autoimmunity is not an attack. It is a misunderstanding. And misunderstandings, even in the cells of your body, can be clarified with the right guidance. You imagine CRISPR as the guide, a soft-voiced teacher who steps into a lively classroom and simply adjusts the tone, helping each learner find the rhythm again. You feel yourself relaxing into this idea. The more you understand, the quieter your thoughts become. It feels soothing to frame the body not as battlefield but as garden, one that sometimes grows too wild, not maliciously, but because it has lost sight of the intended shape. CRISPR becomes the gardener's hand, pruning lightly, encouraging certain branches to rest and others to flourish. You breathe out, slowly, as if matching that rhythm. Then another layer appears, the emotional resonance of such healing. You realize that balance inside the body mirrors balance inside the mind. Autoimmunity, on a symbolic level, can feel like self-doubt, self-resistance, or the internal noise that rises when you push yourself too hard. And so the idea of restoring balance feels meaningful beyond biology. It suggests gentleness not just for your cells, but for your whole being. Healing, in its deepest form, becomes an act of remembering who you are at the cellular level and at the emotional one. The chapter continues unfolding like a late evening sky, cloud by cloud. You imagine quiet laboratory spaces where these therapies are being shaped, not with haste, but with patience. Scientists move with deliberate hands, adjusting the code of life with profound respect. They understand that these cells hold stories, of what the body has faced, of what it has overcome, of what it has misunderstood. Gene editing becomes a way of rewriting the narrative where it has tangled, smoothing the knots rather than cutting through them. You picture a single immune cell receiving such a correction, glowing faintly as if relieved. This confusion lifts. It remembers its role again. You breathe in this imagery, letting it settle. You sense that these therapies are not about turning the immune system off, but about bringing it back into conversation with itself. The term art of balance now feels fully alive. It is not metaphor only. It is practice. The body is always reaching for equilibrium. CRISPR simply helps it find the path. As you float toward the final stretch of the chapter, you sense a question rising. If autoimmunity is imbalance, and CRISPR offers balance, then what lies beyond this restoration? What comes next, when the body remembers its middle ground? The thought unfurls slowly, like a lantern carried toward the next chapter's doorway. You drift into a space where your body's defenses begin to take shape around you, subtle at first, like whispers rising from a quiet landscape. You sense motion, slow, deliberate, as immune cells wander through your bloodstream and tissues. They travel not with urgency but with purpose, patrolling the world within you. Their presence is comforting, like gentle figures moving through a night garden, lanterns in hand, watching over each leaf and pathway. As you follow them, you begin to see how your body encounters the outside world. A single breath brings new visitors, particles of dust, drifting microbes, fragments of scent. Most pass quietly, harmless and unnoticed. But sometimes, something unfamiliar arrives, something that needs attention. The immune cells sense it, leaning closer, reading its patterns the way you might read the grain of wood beneath your fingertips. They don't rush. They observe first, then respond. A virus enters like a shadow slipping under a door. It carries instructions not of your body's making, messages meant to turn your cells into factories for its own replication. But your cells are not alone. Sentinel proteins recognize the viral shape, sensing that something is out of place. They call for help with signals that ripple outward, chemical flares rising into the inner sky. Within moments, other cells gather, forming a protective circle around the threat. You watch this unfolding not as a battle, but as a choreography. Each cell moves with intent, as though guided by a score only they can hear. Some cells engulf the intruder, dissolving it slowly until only harmless fragments remain. Others carry pieces of the threat to specialized centers deep within your body, where instructions for future recognition are prepared. Memory is shaped here, an imprint of the encounter, saved for the next time the same intruder appears. You notice something remarkable. Your immune system is not simply reacting. It is learning. Every infection becomes a lesson, a story written quietly into cellular memory. These stories guide future responses, strengthening your resilience with each new encounter. You feel this knowledge settling into you like soft warmth. But alongside this natural wisdom, you sense something new entering the landscape, a gentle intervention, shaped not by nature alone but by human understanding. CRISPR, the tool borrowed from ancient bacterial defenses, emerges like a clear, guiding light. It does not force its way into the scene. Instead, it appears as a companion, a teacher that shows cells how to refine their recognition. You move closer and see how CRISPR can adjust the receptors on immune cells, allowing them to better identify viruses or bacteria that once escaped detection. It feels like watching a musician tune an instrument, tightening a string here, relaxing another there, until the sound becomes clearer, purer. The immune cells carry these adjusted instructions with them, more prepared than before, more attuned to subtle threats. There is no harshness in this process, only refinement. CRISPR offers the ability to rewrite small parts of the immune story, transforming vulnerability into strength. You imagine its touch as a soft brushstroke, correcting a line on a canvas without disturbing the painting's essence. You follow the CRISPR-guided cells as they travel outward again. Their movements are smoother now, more coordinated. When a bacterial invader appears, round, textured, drifting like a seed, the cells move toward it with a recognition that feels almost intuitive. They know its patterns instantly, as though remembering an old lesson. They respond with a quiet confidence, resolving the intrusion before it grows. The landscape shifts. You drift into another part of the body where infections linger longer, places where viruses hide inside cells, invisible to most defenses. These hidden threats used to cause confusion, slipping past the immune response by blending into the background. But with CRISPR's help, immune cells learn to see them, recognizing subtle molecular hints that previously went unnoticed. It is like giving someone new glasses, sharpening edges that were once blurred. The immune system doesn't attack more aggressively, it acts more precisely, saving energy and reducing collateral harm. You feel the elegance of this clarity, a sense of harmony forming between detection and response. As you move through this internal world, you see examples of infections that once overwhelmed the body, but now appear smaller, more manageable. Some cells are modified to resist viral entry altogether. Others are taught to interrupt viral replication before it begins. These edits are not sweeping rewrites, they are gentle corrections, like smoothing the wrinkles of a page so the story can be read more clearly. You begin to understand that CRISPR's gift is not domination over disease but the possibility of partnership with your own biology. It allows the immune system to do what it already does well, only with more clarity, more balance, more wisdom. In another corner of the landscape, you encounter T cells that have been re-engineered to fight infections with remarkable focus. These cells carry new receptors, carefully designed patterns that match specific viral signatures. They move through the bloodstream like guardians carrying keys that open only the right doors. When they meet infected cells, recognition is immediate. Their response is efficient, gentle, and controlled. Nothing in this system is rushed or panicked. CRISPR's editing has removed the guesswork, leaving only intention. You feel a calm assurance in watching these cells behave, an understanding that resilience comes not from force but from insight. The world inside you shifts again, this time taking on a more luminous quality. You approach areas where infections once left scars, tissues marked by inflammation or damage. Here, the immune system sometimes reacted too strongly, unleashing responses meant for protection but ending in disruption. You sense that CRISPR can help here too, teaching cells to respond with moderation rather than excess. It can adjust the pathways that control inflammation, turning down the volume so healing can proceed quietly. You pause, feeling the significance of this idea. Protection does not need to be loud to be effective. Sometimes the gentlest response is the most powerful. As you drift onward, you notice possibilities unfolding like soft petals. Diseases that once left the body vulnerable now face a redesigned defense, one guided by both natural memory and carefully shaped insight. The potential feels expansive yet grounded, like watching dawn spread without hurry across a landscape. You imagine a future where viral outbreaks meet immune systems already prepared, where chronic infections lose their foothold, where treatment feels less like a struggle and more like a restoration of balance. You picture people receiving edited immune cells that return to their bodies like old companions, wiser and more capable than before. You feel the horizon expanding, not with urgency, but with hope. Near the edge of this inner world, the environment grows quieter. The immune cells continue their steady patrols, their movements confident and calm. CRISPR lingers like a gentle mentor, present but unobtrusive, shaping possibilities without overshadowing the natural intelligence of your biology. You sense that this chapter of the journey is coming to a close, but something in the stillness hints at what lies ahead. Editing infections and rewriting defenses is only one part of the story. Beyond this, larger questions wait, questions about how CRISPR might shape whole systems, whole bodies, whole futures. You breathe into that openness, letting it fill you with quiet curiosity. You slow down now, as though standing at the edge of a quiet clearing. After journeying through the landscapes of cells and memory, of infections and defenses rewritten, you feel a different kind of stillness gather around you. It is not the silence of absence, but the silence of consideration, the hush that arrives when something important is placed gently in your hands. You sense that you are no longer just watching what science can do. You are beginning to feel the question of what it should do. Around you, the world of your inner imagination softens into a wide open space. There are no cells here, no molecular dances, no CRISPR guides moving like careful sculptors. Instead, there is a warm horizon, tinted with the colors of dawn. It feels like a place meant for reflection, as if nature itself understands that the next step of the journey requires quiet thought more than active exploration. You breathe slowly, letting the air settle in your chest. You feel the weight of possibility, not heavy like a burden, but full like a bowl brimming to the edge. CRISPR has opened doors you never expected to see. It has shown you how life can be understood, rewritten, guided with precision more delicate than the smallest whisper. And now, standing here, you sense that each new step forward asks for something equally precise within you, an ethical awareness shaped with the same care as any gene edit. You imagine a scientist at a bench, hands steady, breathing calm, aware that their next decision will ripple far beyond the walls of the laboratory. The tools before them are powerful, but the meaning behind their work is shaped by intention, by values carried quietly in the heart. You begin to realize that science is not separate from the people who practice it. It grows from their choices, their hopes, their sense of responsibility. And in this way, ethics becomes not a barrier but a guide, a soft lantern illuminating the path ahead. As you walk deeper into this reflective space, you sense the many questions that society asks about CRISPR. Should we edit only to heal? Should we allow changes that shape the next generation? Where is the boundary between assistance and alteration, between care and control? None of these questions feel sharp or confrontational here. Instead, they drift around you like gentle currents of thought, inviting exploration without urgency. You picture the early days of CRISPR research, when scientists realized the vast potential hidden within a system borrowed from bacteria. Alongside the excitement came caution, a deep acknowledgement that shaping life requires more than technical skill. It requires wisdom, humility, and the ability to listen to diverse voices. You imagine discussions unfolding like long, thoughtful conversations at dusk, where ideas are exchanged slowly and every perspective is allowed to settle before a conclusion is reached. The path beneath your feet becomes soft, as though woven from all these shared reflections. You begin to see that ethics is not a set of rigid rules but a living practice, one that grows as understanding grows. It is shaped by culture, by community, by the stories we tell about who we are and who we hope to become. And because CRISPR touches the very code of life, it asks humanity to look inward with new clarity. You pause beside a pool of still water. The surface mirrors the sky, but it also seems to mirror your thoughts. You wonder what it means to edit cells with compassion rather than ambition. What it means to pursue cures without widening inequalities. What it means to ensure that the benefits of CRISPR do not become privileges reserved for a few. These questions ripple softly across the water's surface, expanding outward without breaking its calm. You kneel beside the water and watch the reflections shift. You understand that ethical decisions grow from small, intentional actions. From transparency in research, from thoughtful regulation, from collaboration instead of competition, from listening to communities whose lives may be shaped by these technologies. There is beauty in this approach, a kind of care that mirrors the delicacy of the molecular tools themselves. You rise again and follow a faint path leading toward a grove of tall, slender trees. Their branches sway, and each movement feels like a reminder that life is dynamic, always changing. The trees are a symbol of continuity, rooted in the past, reaching toward the future. You feel that same tension in CRISPR, a connection to ancient bacterial strategies combined with the promise of yet unwritten chapters of medicine and biology. As you step into the grove, a soft golden light filters through the leaves. In that glow, you picture the many hopes tied to gene editing, relief from genetic diseases, stronger immune systems, treatments that once seemed impossible. These hopes move like small birds, lifting effortlessly from branch to branch. But alongside them, you notice larger shadows, fears about misuse, concerns about unintended consequences, questions about how to protect autonomy and dignity. You realize that ethics guides us not by eliminating uncertainty but by helping us navigate it. It encourages balance, to proceed with neither reckless momentum nor paralyzing hesitation. To ask repeatedly, does this align with care, with compassion, with fairness? And to keep listening even when answers shift over time. The grove opens into a clearing where soft grass stretches in every direction. Here the air feels spacious. You sense that humanity's path forward is not predetermined. It is shaped moment by choices made in research labs, in policy discussions, in patient stories, in global communities. CRISPR is a tool, powerful, yes, but its meaning depends on the hands that wield it. You step into the center of the clearing and look upward. The sky has deepened into a gentle twilight, layers of color blending like a watercolor wash. It reminds you that transitions can be beautiful, even when they hold uncertainty. CRISPR represents such a transition, one that calls for mindfulness, reflection, and a willingness to honor both potential and precaution. You close your eyes and listen. A quiet understanding rises within you. Ethics is not something separate from science. It is intertwined, woven through every experiment, every edit, every healing intention. It asks us to remain present, to stay patient, to remember that life, at every scale, deserves respect. When you open your eyes again, the clearing feels like a crossroads. One path leads toward deeper innovation, another toward careful regulation, another toward global dialogue. But the most important path is the one that requires all three: innovation guided by care, regulation shaped by compassion, conversation held with openness. You take a step forward, feeling the ground firm beneath your feet. The journey through CRISPR has shown you not only what is possible but what is meaningful. You sense that the next chapter will explore how these possibilities move from concept to reality, from theory to lived experience. You breathe in the last traces of twilight and let your awareness settle softly. In the hush of a late evening, you sense time stretching ahead of you, not as a fixed line but as a soft, unfolding fabric. You picture the future as something gently woven, thread by thread, each strand shaped by discoveries so subtle they might be missed unless you pause to feel their texture. Science, once loud and urgent, now seems quieter, settling into the rhythm of care rather than conquest. And in that slow rhythm, you feel an invitation to imagine how the next decades unfold, not with flashes of noise but with the steady glow of curiosity. The future does not rush toward you. It drifts, like light moving across a wall at dusk. You rest inside that idea, letting it expand around you. The immune system, with all its complexity and elegant choreography, becomes a guide into this imagined future. You see it not as a battleground, but as a living ecosystem, shaped by countless conversations between cells and signals. Each message carries a story, and each story is open to revision. CRISPR enters this landscape not as a force that disrupts, but as a gentle editor, one who listens before altering, one who notices where a sentence of biology has grown tangled and might be eased into clarity. Its presence feels calm, purposeful, almost patient. You begin to imagine how this editor might work in the years to come. Perhaps it helps fine-tune immune recognition, not by forcing precision, but by offering a clearer vocabulary. Perhaps it guides cells like a mentor, helping them remember harmful invaders without overreacting to harmless ones. There is a softness to this vision, a sense of medicine shifting from emergency intervention to steady alignment. You feel your breath match that softness, long and slow, as if the future itself asks you to move gently with it, to let each thought settle before drifting toward the next. In this unfolding future, treatments become less about correcting mistakes and more about understanding why those mistakes formed. You picture researchers studying immune pathways the way a botanist studies a forest, noticing which branches lean, which roots tangle, which leaves change color not out of illness but out of adaptation. CRISPR becomes a tool for tending the forest, for trimming what grows too wild and supporting what grows too thin. It does not impose order. It collaborates with what already exists. It feels like a gardener's touch, careful, observant, respectful of the intricate balance beneath every leaf. As you explore this imagined landscape, you sense how healing begins to feel different. Instead of a battle against disease, it becomes an invitation toward balance. You imagine therapies tuned to the rhythms of the body, arriving like a whisper, a gentle reminder rather than a command. You picture immune cells learning more clearly, responding more gracefully, making fewer hurried mistakes. That possibility feels like a soft light at the edge of your awareness, steady and calm. You let the decades roll forward in your mind. In this future, scientists and clinicians no longer speak solely of curing, but of aligning, supporting, harmonizing. The language of medicine shifts from urgency to comprehension. People grow up understanding their immune systems the way they understand weather patterns, predictable yet dynamic, shaped by conditions that can be read, adjusted, respected. CRISPR technologies, once concentrated in specialized laboratories, become integrated into routine care, as familiar as vaccines or regular checkups. The extraordinary becomes ordinary, and in that ordinariness, it becomes deeply humane. You drift deeper into this vision, noticing how society changes along with science. With greater clarity comes gentler expectations. People speak about health with less fear, more curiosity. Preventive care becomes less about scanning for danger and more about cultivating resilience. You sense how empowering it might feel to know that your body is not fixed in its vulnerabilities, that even long-held patterns can be softened or rewritten with time, care, and understanding. The future feels spacious in this light, wide enough for new stories, soft enough for slow transformation. You imagine communities shaped by this understanding, where conversations about immunity and genetics are grounded in compassion rather than confusion. Children learn early that their bodies are dynamic texts capable of revision. Adults rediscover a sense of agency, knowing that healing can come gradually, shaped by small changes. CRISPR no longer feels like a distant, cutting-edge breakthrough. It becomes something familiar, something woven into the fabric of well-being the way clean water and nourishing food once transformed daily life. Its presence feels integrated, almost seamless. As the imagined decades continue, you notice another shift, a deeper respect for the interconnectedness of all biological systems. You picture healthcare that looks not just at individual cells, but at whole environments, gut microbes, stress patterns, sleep rhythms, relationships, the landscapes people live in. You sense how each of these elements contributes to immune balance, and how CRISPR might support those balances by adjusting what lies beneath them. It becomes less about editing isolated sequences and more about cultivating harmony across interwoven layers of life. You pause within this thought, feeling its depth. Healing in this future becomes a shared act, shared between patient and clinician, between environment and biology, between past experiences and future possibilities. You imagine CRISPR helping to gently lift the weight of inherited vulnerabilities, offering new beginnings that are not abrupt, but unfolding. You picture people living with chronic conditions finding steadier ground, not through sudden cures, but through slow shifts that reshape how their bodies respond. The transformation is subtle yet profound, like the gradual clearing of a long clouded sky. There is a sense of relief in that picture, a quiet hopefulness. You feel it settle within you like a warm stone. The future becomes less about chasing perfect health and more about nurturing adaptable health. You imagine the small triumphs, an immune system that reacts just a little less intensely, a genetic predisposition softened into something manageable, a once tangled pathway becoming clear enough to let healing flow. These are not dramatic victories, but they are deep ones. You let your awareness drift forward until you stand at the edge of this imagined timeline. You sense how the story continues even beyond your view. The future remains open, shaped by choices made with care, patience, and understanding. CRISPR feels less like a tool of change and more like a companion to change, a guide that walks alongside the immune system rather than steering it. And as you breathe into that idea, you feel a quiet gratitude for the possibility of such gentleness. In the final stretch of this chapter, you hold one last image, a horizon lit not by breakthroughs, but by steady clarity. A world where science is woven into life with humility, where each discovery is approached with the same respect you'd bring to rewriting a cherished story. You sense how the dialogue between genetics and immunity continues, soft and ongoing, shaped by both knowledge and wonder. You linger with that horizon a little longer, letting your imagination widen. You picture future caregivers who listen not only to symptoms but to the stories behind them, understanding that each person carries a history written in cells and experiences alike. You imagine CRISPR helping to lighten the burden of illnesses that once cast long shadows, transforming them into manageable companions. Perhaps children born decades from now will inherit not just healthier patterns but gentler expectations about what their bodies can become. You see researchers working in quiet rooms filled with soft light, crafting therapies that feel less like interventions and more like acts of stewardship. You sense the global community learning, slowly and steadily, that progress need not be rushed to be meaningful. Healing can be deliberate, insight can be tender, and as you hold these possibilities, you sense the next chapter forming like a quiet question waiting to be heard. You settle into the gentle quiet that gathers around you at day's end, feeling your breath sink a little deeper with each new cycle. There is a softness here, a kind of patient listening that your body performs without thought. Even as your eyes grow heavy, your inner world remains awake in its own tender way, shifting and adjusting to the calm you are inviting. You imagine your immune cells drifting like small lanterns through darkened hallways, illuminating whatever they pass. These cells do not rush. They wander with a memory shaped by days, months, and years. They notice traces of past encounters, tiny whispers left behind by old challenges, and hold these impressions carefully, as though safeguarding a cherished story. As you breathe slowly, you sense how learning happens even without conscious attention. Your immune system gathers fragments of information from each moment you live, every breath of air, every shared surface, every shift in temperature. It folds these experiences into a flexible record, not rigid like a carved stone, but soft like a page that can be written on again and again. Your body's learning is ongoing, almost musical. Each immune cell remembers a particular rhythm, a series of cues that tell it when to respond, when to wait, when to soften, and when to speak more loudly. These rhythms guide the quiet harmony inside you, ensuring that vigilance does not overshadow rest, and rest does not dissolve into stillness without awareness. You picture CRISPR here, not as a sharp tool but as a gentle editor sitting beside these cellular memories. It reads through the lines written in your DNA, the broad instructions that shape how your immune system behaves, and considers small, thoughtful revisions. Nothing hurried, nothing dramatic, just slight changes that could make learning smoother, responses gentler, and healing more graceful. CRISPR's presence feels like a whisper of possibility rather than a demand. It resembles a skilled calligrapher who sees a letter slightly tilted and corrects only its angle, not its meaning. It works with what your body already knows, honoring the knowledge gathered across your lifetime, simply offering a way to refine what has become blurred or imprecise. You reflect on how your immune system learns not only from battles but from peace as well. The long stretches without illness matter just as much as the moments when you confront something unfamiliar. In these quiet intervals, cells recalibrate, reorganize their understanding, and strengthen their communication. It is like watching a group of dancers return to basic positions to refine their steps. Every night offers a new lesson. While you sleep, cells roam gently, comparing notes, checking boundaries, refreshing the memory of what belongs to you and what does not. It is a rehearsal without tension, a nocturnal workshop that ensures your system wakes with a clearer sense of itself. This constant learning helps maintain the balance you depend on. You think of how children learn languages more easily during sleep, how memories settle like fine dust. Something similar happens here. Your immune cells store patterns from the day, letting the unnecessary dissolve, keeping only the essential shapes. There is kindness in this selection, an instinct to protect without overwhelming you, to remember without causing strain. In this slow night landscape, CRISPR appears again as a possibility woven through future horizons. Scientists imagine ways to help immune cells remember more efficiently or forget harmful misunderstandings. They dream of small edits that correct confusion, strengthen recognition, or ease chronic overreactions. These are hopes shaped not by force, but by a desire for even deeper harmony within you. You imagine one immune cell drifting through a quiet region of your body, carrying a faint memory of an old virus. It keeps that memory like a lantern flame, steady, small, bright. Through CRISPR-based adjustments, that flame could burn with more clarity, allowing the cell to respond sooner if the same intruder returns, or to rest more peacefully if the threat is gone forever. Your body becomes a classroom of sorts, though not one filled with noise or urgency. Instead, it teaches through gentle repetition and careful observation. Each cell learns individually but also communicates its knowledge across a vast network. This connection forms a community of learners within you, each contributing to your health in small but meaningful ways. You sense how rest amplifies these lessons. When your heart slows and your breathing softens, inflammation eases, allowing cells to travel more gracefully. Pathways that were crowded during stressful moments open again. Your internal environment becomes more spacious, creating room for clarity to form. Rest, then, is not an interruption of learning, it is a partner to it. As you drift deeper into stillness, you imagine your bone marrow warm and sheltered, where new immune cells emerge. Each cell is born with a kind of innocence, carrying the ancient blueprint written into your DNA. This blueprint guides its early steps, but the cell will learn much more through experience, adjusting its responses depending on what it encounters in your living world. CRISPR gently enters this picture as a storyteller who can revise the opening lines of that blueprint. By shifting a single sentence, the story of how a new immune cell behaves can unfold differently, perhaps more effectively, perhaps more compassionately. It is a way of shaping the beginning so that the rest of the tale flows with greater ease. You consider how remarkable it is that learning can be written into life so deeply. These small edits do not override nature. They collaborate with it, supporting the biological intelligence that already pulses within you. This partnership reveals a new kind of healing, one that respects the stories your body holds while offering ways to retell them more gently. The night grows quieter around you, and you feel gratitude for the invisible work happening beneath your skin. Each immune cell operates faithfully, even when you are unaware of its presence. You do not have to command anything. Your system recalls its training, adjusts to new information, and continues its slow, thoughtful watch over your well-being. The thought brings comfort. You are never truly alone. Thousands of tiny learners move with purpose through your blood, tissues, and organs. Their dedication forms a kind of companionship, quiet, steadfast, and deeply personal. Every moment you rest, they continue their patient journey, working to keep you in balance. You imagine how future generations might experience this internal harmony even more clearly. As CRISPR technology grows safer and more refined, it may offer ways to correct inherited vulnerabilities or strengthen immune learning from the start. Not as a promise of perfection, but as an invitation toward steadier health and gentler responses. This vision does not rush you. It arrives like a soft breeze, distant yet comforting. You understand that healing is a gradual unfolding, shaped by both biology and experience. Your body does not demand immediate change. It adapts at the pace of breath, of sleep, of seasons moving through you quietly. Your awareness drifts to how interconnected everything is. A single misunderstanding by one immune cell can ripple through your system, just as one corrected cue can restore balance in surprising ways. Learning, then, is not just personal, it is communal. Your cells rely on one another, supporting the whole with each small act of recognition. You picture CRISPR as a companion to this interconnectedness, an artist of relationships rather than an engineer of parts. Its edits guide cells toward clearer communication, helping them distinguish what needs response and what needs compassion. Through these adjustments, your inner ecosystem becomes more coherent, more attuned to the quiet truth of what keeps you well. The more you imagine this internal harmony, the more your body relaxes. Your shoulders soften, the space behind your eyes warms. You feel the subtle shift that comes when your system knows it is safe enough to rest deeply. This safety encourages even deeper learning, allowing your immune network to refine its knowledge with calm certainty. You rest in the knowledge that your body is not merely defending, it is evolving. Each night shapes tomorrow's responses. Each moment of stillness gives your cells room to listen and to remember. Learning has become a form of care, and care has become a form of rest. In this merging, you sense something quiet and profound. And as you drift further into sleep, you feel a final thought rising gently. Your body learns, your body rests, and somewhere between these two movements lies the beginning of whatever comes next, waiting softly in the unfolding dark.